Hello everyone. Let's continue with the question discussion on lipid metabolism. First one is a 5-mark question on digestion and absorption of lipids. First, let's explain on the digestion of lipids. Lipids mainly in the form of triacyl glycerol, cholesterol and phospholipids are entering in our diet. So, the dietary sources are triacyl glycerol, cholesterol and phospholipids. Next, let's explain on the digestion in stomach. Stomach with the help of enzymes like lingual lipase and gastric lipase. The lingual lipase is mostly found in infants because it is helping in the digestion of short chain triglycerides, which is mostly found in milk and milk products, and these infants are mostly depending upon milk. The lingual liquase from the mouth enters into the stomach along with the food, and it is having an optimum pH of around 2.5 to 5. And since the stomach contains hydrochloric acid HCl, these enzymes, since it is having an optimum pH around 2.5 to 5, is therefore continuing its activity in the stomach as well. It is active in the stomach and it is acting upon the short chain triglycerides, and which is mostly seen in milk and milk products. And this lingual liquase is observed to find mostly significant in newborns and infants, who is mostly upon milk. Next one is gastric lipase, and it is also acid stable, having an optimum pH of around 5.4. It is secreted by the Schiff cells and it is stimulated by gastrin, and it up to 30% of the digestion of triglycerides it is helping, and it is occurring in the stomach. Next, we need to explain about the digestion in the intestine. It is undergoing emulsification. Emulsification, that it is the larger particles are dispersed into smaller droplets. The emulsification is prerequisite for digestion of lipids, and these lipids are dispersed into smaller droplets. This is helping in lowering the surface tension, and it increases the surface area, so that the enzymes which is present in the intestine can act upon these lipids easily. This emulsification process is favored by bile salts, mainly by bile salts, and this bile salts is present in the bile. It lowers the surface tension and they emulsify the fat droplets. The emulsification increases the surface area of the particular, and it is helping the enhancement of the activity of the enzymes. Enzymes mainly present in the intestine. What are these lipolytic enzymes which is present in the intestine? That includes pancreatic lipase with colipase, then cholesterol esterase and phospholipase A2. And also this bile is helping in maintaining the neutralizing the acid pH for the activity of these enzymes. So it is also helping in the emulsification process and also maintaining a pH. The bile having a pH of around 7.7 entering the duodenum serves to neutralize the acid kind from the stomach, and it provides a pH which is favorable for the action of pancreatic enzymes. Next, you need to explain about digestion of triacyl glycerols. Triacyl glycerols with the help of pancreatic lipase can easily hydrolyze the fatty acid esterified to the first and the third carbon atom of the glycerol, forming 2 monoacyl glycerols and 2 molecules of fatty acids. Then it undergoes isomerization with isomerase enzymes, and it shifts the ester bond from position 2 to 1. The bond in the first position is then hydrolyzed by the lipase to form free glycerol and fatty acids. You need to write the flow chart, that is triacyl glycerol in presence of lipase getting converted to 2,3-acyl glycerol and then to 2 monoacyl glycerol, that is undergoing isomerase and forming 1. Next, it is getting converted into glycerol and free fatty acids with lipase. Cholesterol ester may be hydrolyzed to free cholesterol and fatty acids, and the phospholipase enzymes produces lysophospholipids and free fatty acids. So that is with the digestive enzymes. Next, we will move on to the absorption of lipids. Absorption, first one, that is long chain fatty acids. They are not directly absorbed. They are absorbed to the lymph, and they forms micelles. Mixed micellar formation is the next heading you need to include, and you have to give a brief description on the micellar formation. Micellar formation, the micellar consists of the digestive products of lipids, namely 2-monoacyl glycerol, long chain fatty acids, cholesterol, phospholipids, lysophospholipids. These are all the digestive products. These are all incorporated into molecular aggregates to form mixed micellars, and it is in spherical shape, and it is having an hydrophilic exterior and hydrophobic interior, and this amphipathic nature. It is having amphipathic nature. Due to that amphipathic nature, the bile salts is helping in the micellar aggregation. Micellar formation is essential for the absorption of fat soluble vitamins like A, D and K. The micellars are aligned to the microvillous surface of the jejunal mucosa, and fatty acids, 2-monoacyl glycerol and other digestive products are passively diffused into the mucosal cells. Next is the absorption of short chain fatty acids. They are directly absorbed. These short chain fatty acids are mostly seen in milk and milk products, and these medium chain fatty acids mostly in oil and mother's milks. They do not require re-esterifications. They can be directly entered into the blood vessels and then to the portal vein and finally to the liver, where they are immediately utilized for energy. They are immediately utilized for energy, and the absorption is also rapid, and the absorption is also better when compared to that of long chain fatty acids. Next, explain about the re-esterification inside the mucosal cells. Once inside the intestinal mucosal cells, the long chain fatty acids are re-esterified to form triacyl glycerols. The fatty acids are first activated to fatty acyl CoA by the enzyme acyl CoA synthase or thiokinase. This needs the lysis of 2 energy compounds are utilized. The 2 such activated fatty acids reacts with monoacyl glycerol to form triacyl glycerols. Majority of the molecules is following this MAG pathway, monoacyl glycerol pathway, and free glycerol absorbed from the intestinal lumen is directly entered into the bloodstream. So, free glycerol is not available for re-esterification, but the cells can derive this glycerol phosphate from glucose by glycolysis, and also add 3 molecules of acyl group to synthesis of triacyl glycerols. Next is the chylomicrons. What is chylomicron? You need to give a description. That is triacyl glycerol, cholesterol esters and phospholipids along with the apoproteins B48 and ApoA are incorporated into chylomicrons. And this chyle from the intestinal mucosal cells loaded with chylomicrons are transported through the lacteals into the thoracic duct and then emptied into the systemic circulation. If the digestion or absorption of these lipids is asked for 3 marks, you can give a brief description with these simple explanations with minor, major digestion, and then passive and re-esterified absorption. If it is for 5 marks, you need to explain about the enzymes as well as in stomach, intestine and various places how triacyl glycerols and long chain, short chain, each separately you have to explain if it is for 5 marks with the diagrams. Next, moving on to the 1 mark question on action of hormone sensitive lipase. Hormone sensitive lipase helps in mobilization of stored fats, especially in the adipose tissue. Hormone sensitive lipase initiates lipolysis by breaking down of triglycerols stored in adipose tissue into free fatty acids and glycerol. Next is a 3 marks question on different types of lipases involved in lipid metabolism and mention its functions. So, if it is 3 marks, at least 3 to 5 lipases need to be explained. Any of these 5 you can study. You need to include the name of the lipase, where it is acting, and what like triglycerides or long chain or medium chain, what it is acting upon, and what are the products formed from it. Okay, thank you.